Good evening, all. Am I audible? Hello, am I audible? Hello. Uh, can anyone respond that am I audible or not?
Yes, sir. We are able to hear you, sir.
Yeah, thank you. Okay, so let's start the session. So, welcome to week 2 of the live interactive session of the Neural Science for Engineers. The course instructor is Professor Vikas V. of Nemhans, Bangalore, and I am the PMRF teaching assistant Vikas Pandit from IIT Jodhpur. This is a brief about me. I think most of you are joining, like you had already joined in the previous class, so you must be aware of it. And I think Sir will also join after some time. Okay. So before going into questions, if you have anything or any doubt regarding the things taught in this week 2, you can ask anyone. Okay. So first thing is that during the class, I will not be able to check the messages. So you all can unmute yourself and respond. Okay, so let's start with problem 1. What is the standard flow for information transfer in the nervous system as discussed in the lecture? Sodium to potassium to chloride, or chloride to sodium to potassium, or potassium to sodium to chloride, or sodium to chloride to potassium.
Sodium to potassium to chloride, I think.
Sodium to potassium to chloride. Okay. Any other answers? Yes, anyone? Paritos?
No, sir, I'm not able to recall this.
Okay. Just yeah. Am I audible now? Hello.
Yes, sir.
Yes, sir.
Okay. Just let this. Yeah. So, any other answers for this? Okay. So, let's. Uh, can anyone recall how the information transfer occurs in a nervous system? Remember, we discussed about action potential in the last class. Then, Sir has discussed more things like inhibitory potential and excitatory potential. Right. So, what are the things that happen for the flow of information in the nervous system? Anyone can recall?
Initially, we have to get activation potential.
Yeah. So,
So for activation potential, we need that threshold 55 millivolt. For that, we can initially sodium will come, and then the potential will change, and then cell from the cell potassium go out, and like that it is maintaining that activation potential.
okay. And what is the role of chlorine or chloride ion? Anyone?
I think it is a gradient voltage.
Gradient voltage. So, what role it will play? This is the complete cycle of action potential, right?
Yes, yes.
So
Chloride will give a in between gradient. So already by default value. So if something comes out again, the potential crosses 55, then activation potential will be there. Otherwise, it will not be there.
okay.
So gradient
So
will determine that.
so what will happen first? Okay. What will happen first?
Then chloride to sodium to potassium. Is it true? Initially, chloride will come and give a gradient. Is it so?
No. Where is the explanation? Just a minute. Okay, fine. Not an issue. Okay. Yeah. Yeah. Any other explanation from anyone? Some part is fine, but what about the correct answer is potassium to sodium to chloride, right? But how is it so? Anyone would like to explain? The action potential part, Sajiji have clearly has explained it clearly, but there is some change. So how the information flow is occurring in nervous system? What is the sequence? Yes.
So, but isn't it the sodium channel that first opens? So, wouldn't it be sodium to potassium to chloride?
Sodium channel first opens, right? So
So,
see the question. It is saying information transfer. It is not explicitly saying that what is this? What you say? It is not explicitly asking for action potential. It is
okay.
saying
Yes, sir.
what is the process of transfer of information or standard flow of information.
Yes, sir. So now I get it. So first, what happens is initially is like when the action potential comes, uh, the sodium channel opens, and as a result of which the previously positive outside layer becomes negative, and the inside becomes more positive. And when it starts its propagation, the potassium channels will open, uh, and as a result of which the inside will again become negative and outside will become positive, and then this keeps on moving to the sodium and potassium channels continuously, and chloride is involved in maintaining the ionic potential.
So is the flow that is shown here, is it right? Will it be potassium to sodium to chloride?
Yes, sir, because they are told it's transfer in the nervous system. So
Hmm.
I misunderstood that we have to start from the action potential.
Yes. So here we are talking about information transfer, right? So it is not action potential. Fine. So the correct sequence will be potassium to sodium to chloride. Okay.
Can you show that explanation? Then we can get some.
Yeah. Also, let me create some blank one. Just
So, but going ahead with the same logic what Niranjana has mentioned.
mhm,
So, if we are having potassium or sodium which is going to start it
mhm.
due to the gradient, and chloride is coming into the picture due to the charge difference.
Yes,
So, in that case, chloride should come in between. No?
yes, but like, okay, let me. Yeah, Devana, you are trying to say something. Hello, Devana. Can you hear me?
Am I audible?
Yes, yes, you are audible now.
Uh, During resting membrane potential, it is said that
Mhm.
permeability to potassium is more.
Yeah,
So, is
okay.
it because of that? The answer is
Yeah,
I am not able to find the
starting
right
is
answer.
right. Yeah.
Okay, okay. And during action potential, permeability of sodium is more. That is known, but the chloride part, I am not very sure of.
Yes, any other explanation you would like to add? So let me explain. Okay. Hello, am I audible?
Yes, sir, yes.
Yeah. So the standard flow of information transfer in the nervous system involves the movement of ions across the neural membranes. So this is clear, right? So the information will travel through ionic exchange through the membranes. This is clear. Any like, is there any doubt in this? This is something I think everyone is clear, right?
Yes, yes.
Okay. So the process begins with the movement of potassium ions, K positive, out of the neuron. So we have, I written, can you see at the bottom? Yeah, let's say this is, uh, let's say it is outside, or, mm, okay, I think here we can draw. So let's say, I am not drawing that clearly. Okay, if this is inside of a neuron and this is outside, okay, so first potassium ions move out of the neuron, okay, followed by movement of sodium. So there will be a sodium gate that, I think I can put it. Yeah. So sodium gate. So then this movement occurs. Then sodium moves in the neuron. Na positive goes inside the neuron. Okay. And finally, chlorine chlorine ions move into or out of the neuron. Both sides can happen to neutralize, to make it again to minus 70 millivolt. That is the resting potential. This sequence is essential for generating the action potential and transmitting signal across neurons. Okay, so that is why this potassium to sodium to chloride, this is the representation. Clear?
Okay.
Okay, so first sodium will move. Sorry, not sodium. So first potassium moves, that will move out. This will go out. Sodium will go in, and then chloride can move both sides. It can either go in or out depending on the charge neutrality. Like where it is more, is it negative or positive? It will try to balance it and make it to the resting potential of minus 70 millivolt. Okay, so let's move to the next question. That is, what is the term used for local changes? Remember here again, we are not talking about global changes. We are talking about local changes in membrane potential caused by movement of ions that do not result in action potentials. So something that is not resulting into action potential. that means it will be below minus 55 millivolt, right? It will be less than minus 55 millivolt. So what is it called? Is it synaptic potential, hyperpolarization, action potential, or inhibitory potential?
It is inhibitory potential.
Synaptic potential.
Okay, and synaptic potential. So now let's discuss why inhibitory potential or why synaptic potential. Yes, Sajiji, can you explain why you think it
Uh,
is?
Actually, whenever the action potential is probability will be reduced by inhibitory potential. That means and hyperpolarization is amplification. Actually, there is a chance for more action potential. So I think inhibitory potential is will not result in the action potential.
Yeah, and I think Devana, yeah, you said it is synaptic potential. Yes. So why synaptic potential?
It is a localized potential which is non-propagatory.
Mhm.
So yeah, most of them tend to be depolarizing. So I thought it is synaptic potential.
Yeah. So they actually it is not explicitly saying what kind of potential. Will it inhibit or will it be inhibitory or excitatory? And these occurs at the synapse. That is why they are called as synaptic potential. So this one is also partially correct. I will not say fully incorrect, but it includes inhibitory as well as excitatory potentials. Okay, so both together are called as synaptic potential. Clear?
Okay. Okay.
Yeah. So it we are talking about two terms, EPSP and IPSP, and these both are occurring at the synapse, and these together are called as synaptic potential.
But one more thing, sir, excitatory things will make action potential actually.
No,
Inhibitory, it is diminishing that chance.
it
So I
will
think
diminishing. Yeah.
so we are they are asking why it is asking clearly that do not result in action potential.
Yes, they do not result
Then,
in action potential.
so in
So,
other case, excitatory may have start action potential.
but if
So
excitatory becomes beyond that, it will not be called as EPSP. It will be called as action potential. If it goes beyond the threshold, right? You say that it is minus 70, right? Minus 70. I am not putting the unit. Let's say this is minus 55. This is the threshold, right? Then let's say 0. I am not drawing to a scale. Fine. 30. So if it is like this, it will be EPSP, right? You are saying this.
yes, yes.
Yeah. But if it, let me put one other color. Yeah. So you are saying that if it goes like this, beyond this, it will result in action potential,
Yes.
right? It will,
Yes, sir. Yes, sir. Yes,
it
sir.
should go directly like this, and
There is
then,
a chance to get action
yeah.
potential.
But
But in previous case, inhibitory case, there is no chance.
there is no chance. Yes, certainly. In case of inhibitory, when we are going down, there is no chance that there will be an action potential. But in case of excitatory also, there will be no chance because if it is going beyond minus 55, the stimulus is too high, it will actually be called as action potential. And the question itself asks which do not result into an action potential. Okay, it is more about term. It is both inhibitory as well as EPS. Like these both are together called as synaptic. Fine.
So can you explain the last statement again? I had the very same doubt that Saji sir also has.
yeah, that if you get this EPSP beyond minus 55, like the difference is I think 15, right? If it is more than 15 volt, so what will happen? Like then it will actually return to Xn potential, right? This is what you are asking, Paritosh?
Yes, sir. Yes, sir.
Okay, so will it then be called EPSP? If
No,
it
in
is,
that case, it will be called action potential.
yeah, it will be called as Xn potential. So that is what I am saying, that EPSP and IPSP are those things that will not result into Xn potential, right? And these together are called
Okay.
as synaptic potential. So if we are saying inhibitory potential, that is also partially correct, but along with that, it should be excitatory for synaptic potential also, right? These together will form the synaptic potential. Fine. So synaptic potential will be the correct answer. Okay. How does IPSP affect the chance of an Xn potential? So I think this is in continuation with that. So any answers? Will it increase the chance?
Decreases.
It
Decreases the chance.
decreases. And the name itself, it is inhibitory, right? Inhibitory in nature. It's this one, yeah. So it will decrease the chance, reduces the chances of an Xn potential from occurring downstream, not there along the stream. It will affect. Yeah. So what is the significance of
So
EPSP
downstream, what does it mean?
downstream, in the sense that if, like you must remember from the lecture, that if suppose here uh, EPSP occurs, um, okay, so this, since it is a change in potential, right? So last class also we
Got
discussed,
it. Got
when
it.
there is a, got it, right? Whenever there is a uh, change in potential, it will propagate, and that we can say as a current if we talk in terms of engineering or electronics. Okay, so this propagates downstream. Okay, so what is the significance of EPSP and IPSPs in the nervous system's computational process? So remember, when we were talking about action potential, it was about transmission of stimulus or information. And when we are talking about EPSP and IPSP, they are computational, okay, where they are manipulating the uh, existing, you can say the information is being changed or manipulated. Okay, so they are the primary currency of the nervous system. They modulate the generation of the action potential. They facilitates the action potential's uniform transmission, or they form the structural component of neurons.
B.
B.
They
They modulate
modulate the.
generation of action potential. And how?
By increasing or decreasing the potential values, and subsequently that will affect the action potential formation of action potential.
They modulate, because we are saying that they are modulation means they are changing the, or they are controlling the generation of. So suppose an action potential was about to happen, but if an IPSP occurs, it will not, it might not let the action potential happen. And suppose an there was a stimulus that was not going to result into an uh, a action potential, but if EPSP has occurred, so what will happen, that it will force the small signal also to amplify and result into an action potential. Fine, is it clear?
Yes, sir.
Okay. So what is the significance of the membrane potential reaching minus 55 millivolts in neuron? Does it represent the resting membrane potential? Is it the threshold for triggering an action potential? Will it indicate the peak of an action potential, or it denotes the maximum potential for EPSP?
It should be B. Whatever doubt in the D option is there, because in the second question we have discussed that if EPSP is reaching just
Minus
beyond minus 55, we won't be calling it as EPSP.
EPS.
So it should
Yes.
be the maximum of EPSP as well. So I guess
Yes.
both B and D would be the right
Yeah, B
answer.
and D also we can say is somewhat correct, right?
Yes, sir.
Yeah. Yes. Any other answers for this? I think, yeah, Niranjana, are you there?
Yes, sir.
Yeah.
Uh, I just feel like B is a more apt option, though both of them sound right. B sounds
Yes.
to be more apt.
Yeah. So let's say that we are not going into multiple and like where multiple answers are correct. Uh, from next time, I will try to have such questions also where we can discuss more. But here, I think let's go for this lecture. Let's go for the most appropriate or the best answer. Okay. So by that logic, we can go for B, and D also will be partially correct, right? But the most apt answer will be B. Is it fine? Any questions still here? Yes. Anyone?
No, sir.
Okay. So, what term is used to describe the phenomena where an action potential is either generated or not generated based on stimulus intensity? So, before going into this, remember one small thing that we said that for an action potential, what was there any change in amplitude?
No.
Yes.
There was no change in amplitude. But here we are saying that an action potential is either generated or not generated based on stimulus intensity. So if the stimulus is less than a threshold value, it will lead to no action potential. And if it is beyond this threshold value, it will lead to full action potential. There, whenever action potential happens, it is the full height of the action potential. The amplitude remains the same. So what is the term used to describe this phenomenon?
All or none.
All or none phenomenon.
All or none phenomenon, right? Okay. In context of neuron activation function, the sawtooth graph. Remember this graph? Such a graph was there, right?
Tan H, I think.
Which one, sir? Hello?
It is similar to tan H.
Tan H. Okay. And what is the structure of tan H?
It is little, I cannot draw it. Not sharp,
Yeah,
but yes,
like
like
this,
that.
right? And any other answers for this? And what is sigmoid curve?
Sigmoid is
S
used
shape.
for, I think, in artificial intelligence.
Yeah. Yes, Devna, you are saying something?
S shaped. That is sigmoid.
S-shaped. Uh, like this?
Yes.
Okay. And what about rectified linear unit? Yes, anyone? Or linear? How linear curve will look like? Yes, anyone?
A straight line with an offset from either the x or y axis.
Yeah, something like that, right?
Yeah.
So, which one is the best one that resembles this?
So which one is for rectified linear unit?
Yes, the rectified linear unit is actually this one.
Okay.
Okay, so tan H will also resemble, but not perfectly. It is somewhat, you can say there is a gradient, right, Sajid? Hello.
But
Yes.
in this lecture, he says that it is resembles damage.
Yeah, it resembles most closely to tan H, but the perfect resemblance will be rectified linear unit. If we say directly in terms of ANN, tan H with we can say with some parameters change will actually all result into ReLU. Okay, that is a modified form of tan H. Okay, so this is, we can say, similar, but this one is the most correct. Okay, which is commonly used in case of ANNs. Fine.
So what are ANN and CNNs? I'm
Uh,
not from this background, that's why I don't know.
yeah, it's just like you have central nervous system, right? That you are aware of, right?
Yes, sir.
CNN. So when in sense of computers, when we are trying to replicate neural networks, we are calling it as artificial neural network. Okay.
Okay.
So I think going forward in the course, you will be introduced to it slowly.
Okay. Thank you.
Okay. Yeah.
Sir,
So these,
ReLU is not like it is linear only. ReLU rectified, it is almost linear kind of slope.
yeah, almost linear, but like we can say somewhat like this, where we have 0 and then 1, but in between there will be a linear slope, right? It will be a rectified function of the linear slope.
But
Uh,
I think TANH is more appropriate.
yes, but it have more curves if we go properly in tan H. Okay,
Okay, sir.
let's like in the next class I will try to put both the, not both, all of this, and then we can like we can approximate which one will be the better, right? Fine.
Okay. Okay.
Yeah. Actually, tan will have you can say both positive as well as negative, right? Can say ReLU will be like 0 and 1s. Okay, we will discuss in the next class, no tension. I think then I will be able to explain better. Okay, so yeah, what is the role of Manning in the propagation of the action potential? Mailing. First, what is this mailing? Anyone remembers what is this?
Myelin is this one housekeeping cells work.
Yeah,
That's what they say.
yes.
Lecture we say housekeeping. But it
Like
increase, it decreases the It insulates, mainly insulation, and speed up the transmission. That's what he's saying. It is
Yes.
housekeeping people.
Yeah, that is a term given by sir. But if we talk in terms of structure, I think Paritos must be knowing. Or yeah, Devana, what is your background? I think you didn't join in the previous class.
Yeah, I hadn't, sir.
Yeah,
It
so
is
yeah,
secretion from glial cells,
okay.
which covers the neurons.
Yeah, that is fine, man. But I was asking, what is your academic background, like what you have studied?
Uh, Actually, sir, I'm from medical background only.
Okay. Yeah, that is why I was asking because
Correct,
we have
correct.
we have different variety of people here.
Correct, correct.
Someone is from medical background. They will be knowing more in details about this.
Yes, sir,
So,
yes, sir.
what I understood is that it is a fat. Is it correct?
Sorry, sir.
It's a fat. Fat uh, in case of making chemical structure, it is a
Yes,
fat. Is it
yes,
right?
sir. Protein and lipids also. Yes, sir. Mainly
Yeah.
proteins, I guess. Sir.
Okay. Okay.
Sir.
So what it does, it will it enhance? I think Sajiti have already answered some part of it. So will it enhance the membrane potential, increase the resistance of neuron, or insulates and speeds up the transmission of action potential, or decreases the diameter of the neuron?
C, sir. It insulates
See,
and speeds up. Yes.
insulates so that the you can say that action potentials will jump instead of going in a straight slow linear fashion. They will tend to jump, right?
Yes. Yes.
And if we talk in terms of like electronics or electrical, they will act they act as low resistance part where they tend to transfer the signal from here to there. Fine.
Okay, sir.
So, which part of the neuron is responsible for local changes in current that lead to generation of action potential? Here, remember, we are talking about local changes in current.
Nodes of Ranvier.
Nodes of
Yes.
Van Weer.
Generation.
Yeah. And any other answers?
I think it is same. It is starting from
Nodes
the
of
action and lots of one.
Hmm. So,
Action terminal both are same.
hmm. So it is the part that is
Ini.
responsible for this uh, change is actually the axon. If we are talking about the cell, okay, the neuron cell dendrites. I'm not that good in drawing, so I'm just drawing like this. Yeah. So let's assume this is axon, though it does not look exactly like that. So see, the question is which part of the neuron is responsible for the local changes in current that lead to generation of the axon potential. So this small local changes that leads to generation of the axon potential. So it is the axon that is responsible for generating axon potential through local changes in current. And
End of axon is known as this one.
what
No,
Axon.
you are saying?
initial part of the axon is
Axon
called
is
as
known as
axon.
action.
Yeah, what you are saying? Yeah, initial part of axon is known as yeah,
Initial segment of the axon, which would be the more apt
y-
answer.
yeah, more apt answer. So like we cannot say the whole axon part, the starting
Whole of
part.
the
Yeah,
axon.
yeah,
Yes, yes.
yeah. So why is it
Answer is
necessary?
what, sir? Answer is
Axon answer is axon.
It may be starting of and action, no, that is nodes of runway or action terminals.
Axon terminals will be both terminals, right? This and this.
Actually, it is said that the concentration of sodium channels is much more in the initial segment, the starting
In
part of the axon.
this part, right?
Uh, yes, so that's why the generation of action potentially seen over there.
Yeah. So we can say that the one side of the axon, right?
Yes, sir. The starting part, just before the
Starting.
nodes would start, just before the myelin would start.
Okay, so if I would have put here like in case instead of axon terminals, if I just put terminal, I think that will be then this will become the correct answer, right?
But
Node,
generation of no, I don't
nodes
think
of
so.
runway is the starting point of action. No?
No, no, that is
No.
between this
No.
where we are having this
Two insulations, two
Malin, yeah,
parts of
two
insulation.
insulin. So this is the node of Ranvier.
The
This
gaps in
part,
between are
the
called
gaps
as
in between
nodes.
where we have this membrane channels.
Appropriate answer may be action terminal only.
Yeah.
Like,
Okay. This one is something like from taken from the lecture. Why is it necessary to understand the structure of nervous system? So, is it purposefully to make the course more challenging, or to appreciate the beauty of online education program, or the structure is essential for grounding the knowledge of the nervous system, or we are just reading it because it is irrelevant, even though it is irrelevant.
Just kidding. As an engineering student, to make the course more challenging,
No,
it is C.
yeah,
it's
but challenging itself is very new to us. We are aerospace engineers. I'm basically study
yeah,
aerospace. It
yeah.
is
So
studying this itself is a big challenge. What he says is correct.
yeah, certainly, like I am also from space science, and I am learning it. For I am also learning this part where this biology and this thing is uh, concerned. But yes, the yeah, other part, the electronics and this, that is something I already know. Yeah. Yes, David.
So can we be asking doubts now, or should it be in the end, sir?
You can ask, not an issue.
So, like regarding the cell membrane,
Mm,
I've read it somewhere that cell membrane's function is compared to a capacitor.
mhm,
So, so how is it like actually I don't know how capacitors work.
mhm. So
So, how is
uh,
the analogy made?
you you can say that when we are talking here in terms of when we are talking especially about neurons. So here, though it is ionic current, so if we take the analogy to a normal current, right, where electrons are flowing instead of ions, electrons are flowing, so that we consider as current, right? From like
Yes,
in the
yes.
last class also, I was saying that there must be difference of potential. Say this is plus V, one I can say, and this is minus V, two. Okay, fine. So there is a difference of potential, then there will be a flow of current. In this case also, when there is a difference of potential of ions, ions are flowing. Then there are two, three components if we say in terms of electronics or electrical. First and the most basic is the resistance. So I think this you are already aware of, that it will a component that will resist the flow. Like if there is something that will try to uh, not stop but just slow down the flow of electrons, that we call as resistance. Then there is a capacitance, so which actually tends to get charged. That means it will try to, if a current is going into it, we are talking about DC current, let's not complicate it with AC and all. So it will try to store charge and get charged. So the current that is going through it will not be allowed to move easily. It will just tend to get itself charged and remain there. Are you getting some idea about it? So this is something that stops, capacitance is a component that stops the flow of electrons. Stops, not just
Okay.
slows down, it not just resist, it actually tries to stop up to a potential, like up to its charging capacity, it will try to stop it. And once this is removed, the current is removed, actually it will try to discharge and it will tend to act as a voltage source. So it will itself try to make the electrons flow once this pressure is removed. So you can say the cell membrane actually stops the flow of ionic current, right? It will not allow direct flow of the current until unless it is tuned or it gets stimulus. Is it
Correct.
true? Yeah.
Yes, sir.
So,
Yes, sir.
so we can understand
Otherwise, one way is that we can say that it is one side is one potential, another is another potential.
otherwise.
In
Yes,
capacitance also, one side is plus, one another side is maybe negative. So
negative.
there is a difference in the potential can keep in capacitance. The same way,
Yeah.
a membrane also doing the same function.
Same. Yeah. It is ensuring that these two, like the right hand side and left hand side, are at two different potential, just like cell membrane, where inside and outside have two different potential. Fine.
Diodes are also supposed to be like that, no, sir.
No,
I'm very poor in
di
this. Okay.
no, no, not an issue. Diodes have again a different function. They, you can say, in normal cases where if we have a diode, okay, a component, and we are connecting it into one direction, say the current is flowing like this, it will directly flow. But if it is no, if we try to flow the current in the opposite direction, it will not flow. So you can say it is diode is more like a unidirectional switch or unidirectional, uh, what you say,
Channel,
oh,
this people
channel.
will
Yeah,
channel.
yeah, unidirectional channel. So it is more like a unidirectional channel where only one directional flow can occur.
Okay. Okay.
Okay? So most of the natural thing, even the air, is modeled using resistances, capacitance, and inductances. So in case of electrical system, when we are studying, we channel everything as a combination of resistance, capacitance, and inductance. So that is why here also you will see some models like that.
Okay. Okay.
Okay. Yeah. Any other questions? Oh, so let's move forward. Yeah. What is the role of neuron in the nervous system? Neurons produce myelin, or are they responsible for housekeeping tasks? Neurons serves as the fundamental computing unit, or neurons regulate calcium channels.
Fundamental.
Yeah, what? Which one? Fundamental
See,
computing unit. Yes, see. So they do the computations, and they are considered the fundamental computing unit of our nervous system. Right? So all the why we are calling it as fundamental computing unit, fine, but why fundamental?
Like if you say in computer, fundamentally it's a bits and bits, no? One pulse. So
Yeah,
this also gives one pulse sound at a time.
yeah,
So we
and
can say it is a fundamental 100 like the binary. You can say.
binary. So actually, the computation is occurring at even a scale lower than this. We can say that EPSP and IPSP, they they are actually yielding this computation, but they on their own cannot result into a complete computation. A neuron is something that will lead to the inside it. all the computation like EPSP, IPSPs will be there, and then the signal that comes out, that is the modified one. Calls, yeah, okay. Just a minute, let me disconnect this.
I think this adding and subtraction is doing with two
Yeah,
channels, dendrites.
yes,
So neuron is only doing the first computation, adding.
yeah,
Branches, power gradients are coming and it is adding. That's what sir was saying.
yes.
So adding is doing done in this
Hmm.
levels, neuron system.
Yeah. So it serves as the fundamental computation unit. Just a minute. Let me just disconnect the calls. I'm not sure why is it. Just, yes. Yeah. Can we move forward? What is the significance of axon hill lock in the generation of axon potential? It is the endpoint of the axon in terms of generation of axon potential. It is asking, okay. It determines the location of synapses, or it integrates the signal and is the origin of the axon potential, or is it that it produces synaptic vesicles? Yes, please.
C, C.
See, it integrates signals and is the origin of axon potential. Any other answers?
Same answers.
An answer. Yes. Anyone can explain or remembers?
I
What
think
is
it is end point of the axon. Axon, this end is known as axon hill.
no? It is if we say I'm not drawing the dendrites. Let's say I'm just drawing the axon, right? And let's say that. So this area will be axon hill log. Am I right?
Yes, sir. Yes, sir.
So this area of the area, not the other one.
Must be C, no sir.
Yeah. It its role is the generation of axon potential, and it integrates the incoming signal from the dendrites and cell body of the neuron. So some signals are coming from dendrite as well as like cell body. Cell body will do some computations. We learned that incoming signals come from dendrite, or and outgoing signal, which part gives out the signal to the next neuron?
Axon terminal.
Axon terminal. So the flow is like this, uh, that it goes in, then some computation is occurring at the cell body, and if the computation results some signal beyond the threshold of our minus 55 millivolts, say, then an axon potential will be generated at this axon hill. Fine.
Yes,
And
yes.
it will then generate towards the synaptic transmission part, that is towards our synapse. Okay. So how do chemical synapses differ from electrical synapses? Electrical synapses involve quanta packets, or chemical synapses convert electrical activity into chemical activity. or electrical synapses result in release of neurotransmitters, or chemical synapses involve low resistance pathway. So what are the difference, or what is the difference?
Option B, sir.
Option B,
Chemical synapses convert electrical activity into chemical activity.
chemical activity. So
Mm.
can you try to explain it further? Like,
Yes, sir. So basically, the chemical synapses are the ones that release neurotransmitters. So like when the action potential is like propagated throughout the neuron, it signals for the release of neurotransmitters through the synaptic vesicles. Whereas the electrical synapses are actually closer. The neurons are actually closer. So
okay,
there's just the movement of charge as is without any neurotransmitters in
yes,
use.
yes. So that is the main difference, and we can say that the chemical synapses convert electrical activity into chemical activity. Fine. And these neurotransmitters are they released from electrical synapses? I think you have already answered it, right? They are released from which synapse?
Chemical synapse.
Chemical synapse, right? Just- And uh, which type of synapse involves low resistance pathways?
Electrical.
Electrical. Okay. So we can say that these are actually just reversed. Okay. What is one of the important functions that occur at a synapse? Blood circulation, synthesis of transmitters, amplifications, or muscle contraction? Yes, anyone?
Synthesis of neurotransmitters.
Synthesis of neurotransmitter, right? So, sorry, yeah, put that correct. Synthesis of it is not saying generation, it is saying synthesis. It is actually amplification. Even if at the synapse, you say that, how can we explain it? Yes, anyone aware with this amplification process? How is it happening?
Synapse also acts like a receptive zone where again EPSP or IPSPs get computed, like either get added or deleted.
Mhm.
Signals get added or deleted.
Yeah,
So synthesis of neurotransmitters happens at the soma in the cell body of the neuron itself.
yeah. So it is
So
not at the synapse, right?
yes. There only release of the neurotransmitter in the
Uh,
synapse. So synthesis actually happens in the cell body.
cell body. So amplification is something
Yeah.
that happens here, right?
Yes.
And
Yes.
it is not the
How
synthesis.
it is happening? How is happening? Can you little bit elaborate?
Uh,
Which
like
one? Yeah.
synapse. Just like we had learnt about IPSP, EPSP. So it can receive many IPSPs or a few EPSPs or many EPSPs and few IPSPs. So depending upon that, if EPSPs are more, then amplification of the signal happens and it is converted into action potential. Uh, if there is more of now terminals and plate in the sense of here, so you see more of amplification.
So we can say manipulation like IPSP and EPSP. and here mostly EPSP, right, is something that is responsible for amplification.
My question is, amplification means something has to be amplified. Amplitude is amplified or speed is amplified.
We
There
can
is
say
some
that
magnitude is
Just frequency, I guess.
stimulus.
frequency is increased, then that is amplified. So
No, it is mostly stimulus. If there is a small stimulus, actually, which was not capable of generating the action potential, if we have an EPSP, it will result into an action potential. So the EPSP will assist the stimulus, like it will increase the intensity of stimulus to result into an action potential.
that way, IPSB is negative for amplification.
Yes.
So, be possible. So, amplification is not correct.
Yeah.
Yes,
Sometimes. Yes. Peritos. Yeah.
clubbing whatever has Davana ma'am has mentioned and Sajid sir has mentioned, like it can either amplify, it can remain same, or
Or it can decrease also. Yes.
yeah. So according to this option, it's right.
Yeah. And it is being considered as an important function because like that will result into detection of small stimulus also. Like we can say that if you are giving a very small stimulus, it is being detected. That is something of relevance, I can say. If we are giving a big stimulus and that also is not being recorded or not being processed, that will I like that will not result into something like where you are going to process it. In fact, you are going to ignore it, right? IPSP will result into may result into ignoring a stimulus that was actually relevant. Yes. Anything on this? Hello. Can we move forward?
Yes, yes.
Yes, sir.
What ensures the specificity of a neurotransmission action at a synapse? So specificity, we are saying that means specific to one particular thing, right? So what is that the property that ensures the specificity of the neurotransmitter action at the synapse? Is it random diffusion of neurotransmitter, varying receptor configuration, uniform amplitude of the action potential, or specific receptor for each neurotransmitter?
Specific receptors for
A specific receptor that this neurotransmitter will bind with, right? So, like acetylcholine act only on a specific receptor, ensuring specificity, right? So, for each neurotransmitter, there will be specific receptor that will only respond to it. Or sorry, it will only act upon. Fine.
Sir, I have a question, sir.
Yes.
The previous one, why can't be option B also? I do understand that B seems the most right, sir, but not varying receptor configurations also contribute to it.
We can say each receptor will have a different, not configuration, we can say structure, right? That it will fit into. You might be remembering the lock and key mechanism, something where receptor and this transmitter bind together and these things, right? So this one, I think it is more about when we are talking about varying receptor configuration, that means like more than one receptor are configuring together. What like I put the option thinking that okay, and when I am talking about specific receptor, I am not going into the chemical configuration. Certainly, chemically they might be very similar, but if we just see it like as an entity, they are different. So I kept it. Is it clear?
But I'm not still convinced.
Okay. So, like when we are talking about configuration, what do you understand configuration?
Either changes in structure or maybe some kind of storage mechanism,
Okay.
something of that sort.
So you are saying when we are talking about configuration, that like you include configurations, structural and chemical configurations into it.
Yes, sir.
Okay. In that case, certainly it will be true. Right. But what the opposite condition in overall configuration is different.
Okay, sir. So
Like
my question is like, aren't there like any receptors which just have varying configurations, or is it the reason for the answer being only, or
no, I'm not getting your question. Yeah.
I'm just asking like these varying receptor configurations, do they don't exist
Varying
like
risk,
in one
no,
for your answer?
they will exist. Sorry.
Okay, sir.
Like they,
I think different variety of receptors are there.
yeah.
It has some kind of functions. I think one can explain more specific. So one can give only kind of one. I think.
They are mostly like what I know is that they have some like lock and key mechanism, that one kind of neurotransmitter will be specific type of terminal. It will not fit into any other. Yeah, I think someone bells you now. Correct. Hello. Yeah.
Even if the receptor configuration is different, it should have its specific neurotransmitter which can go and bind to it. So that's why D would be a very like proper answer for this
Proper.
question.
Okay. Okay. So there are receptors that have different configurations. The same receptor with different configurations.
Yes, yes. Depends on the subnets it is made up of.
Okay. So the chemical units you are talking about.
Yes, yes. Receptors are again they are made of various elements like proteins,
Okay.
phosphates. So
And mostly like what is there on their active sites, we can say the active sites
yes,
will differ.
yes, yes,
So
yes.
yeah. So if when we are talking about this, let's not when I'm putting this option, I was not talking about varying configuration in terms of active sites and all. I was just thinking
Mhm.
about different the receptors having different not receptors, but you can say combination of different receptors. But yeah, if we are talking about the configuration, it is a confusing option, I say. I'll continue this. Yeah. Yeah. In then is it clear now?
Yes, sir.
Okay. In a neuromuscular junction, what triggers the release of calcium leading to muscular activity? So when we are about neuromuscular junction, that means a neuron connecting to a muscle, right? Muscle tissue or cell, we can say, right? So what triggers the release of calcium leading to muscle activity? Is it synaptic vesicles, acetylcholine, action potential, or sarcoplasmic reticulum?
It's acetylcholine, sir.
Acetylcholine. So it is that causes the release of calcium, which in terms leads to muscle activity. Just try to revise that part where see acetylcholine is the receptor. Remember, I think in the previous questions also we discussed it is it is the neurotransmitter that will act on specific receptors. Here we are talking about the release of calcium. Okay, so that calcium release as a part of the action potential to maintain the nuclearity level. Last part where we have to come to the action potential in the action potential, that last part, it is